Hello guys, I am Shubham from Show Tutorials. We are starting with the new series of CSIR NET pyqs. Okay, so first we will be seeing June 2025 paper. Okay, full length papers हम देखेंगे. So this is the first video of June 2025 part B. Okay, part B. In every video we will be seeing 10 questions. Right? हर वीडियो में 10 questions होंगे from this series. So before we start, subscribe to our channel and press the bell icon to get notified whenever new videos out. This is absolutely free from your side, but it will help us grow. Let's see the first question. Which of the following chemical modifications of uridine residue occur after transcription of tRNA? Okay, chemical modifications of uridine, right? So basically molecular biology से question है. मोस्ट ऑफ द क्वेश्चंस आई विल बी कवरिंग जो एरिया हमने ऑलरेडी डिस्कस किया है, लाइक मॉलिक्यूलर बायोलॉजी हमने पूरा स्टडी किया है. यू विल फाइंड द प्लेलिस्ट एज वेल एज जेनेटिक्स, सेल बायोलॉजी हमारा चल रहा है अभी, एज वेल एज बायोकेमिस्ट्री चल रहा है, एंड along with techniques based question which are easy to solve. Okay, the questions those are easy to solve, that also I will be showing you that how you can solve quickly. Okay, with basic idea also you can solve those questions. So now we chemical modifications of uridine. If you don't know about this, I will be giving you this chart, very important. Okay, so I will give you like small informations also in this playlist, which is very direct answer you will find from the table. Okay, see now, इसमें uridine है. Okay, uridine. So uridine में ribothiamidine है, dihydrouridine, pseudouridine and 4-thiouridine. pseudouridine तो हमें पता है, राइट, टीआरएनए में हमें मिलता है ये pseudouridine. इसके साथ साथ राइबोथाइमिडीन है, एंड रेस्ट अदर्स यू कैन रिमेंबर, राइट. So huge amount of repetition is seen in CSIR NET. So this figure is of all 4 bases in tRNA can be modified. मतलब जो भी बेसेस हमें मिलते हैं tRNA में, एडेनाइन, गुआनिन, साइटोसिन, यूरासिल, सबका मॉडिफिकेशन पॉसिबल है. ओके, सो हियर इट इज रिटन सी some of the more common modified bases. Modifications of pyrimidines, cytosine and uracil are generally less complex. Complex कम है than those of AG, purines, right? Purines का complexity modification का high है compared to pyrimidines. Got it? तो इसका आंसर होगा, see the options, ribothiamidine and pseudouridine. Yes. तो फर्स्ट ऑप्शन ही आंसर है. Got it? N3 ये हमें नहीं दिखने को मिलता, राइट. pseudouridine is seen but not the other options. राइबोथाइमिडीन जो ऑप्शन 2 में है ये भी हमें मिलता है बट नॉट N3 मिथाइल यूरिडीन. तो फर्स्ट ऑप्शन इज द करेक्ट वन. नेक्स्ट क्वेश्चन, दिस इज ऑल्सो वेरी इंपॉर्टेंट कॉन्सेप्ट. एक्टिवेशन ऑफ CDK2 in vertebrates is dependent on. So this is from the cell cycle. Okay, cell cycle अभी तक हमने डिस्कस नहीं किया है, बट दिस इज ह्यूज अमाउंट ऑफ रेपिटेशन इज सीन. बहुत बार इसके रिगार्डिंग क्वेश्चन आया है, तो इसका भी टेबल है, राइट. दिस इज वेरी इंपॉर्टेंट, यू कैन मेक अ स्क्रीनशॉट ऑफ दिस. cyclines. So these are the 4 cyclines. How you can remember this? I will tell you. Remember the word deer. Okay, deer. R को रिप्लेस करना है with B, right? So these are the cyclines. 4 types of cyclines है, and CDKs are also 4 types. 4, 6, 2, 1. Okay, now इस टेबल में देखो जो साइकिल के फेजेस है G1, S, M. तो G1 CDK D and 4, 6 पार्टनर है. E का 2 है. ओके, राइट. एंड S CDK A है साइक्लिन CDK 2 and 1, and M CDK cyclin B CDK 1. Got it? Remember this order I have showed you. Got it? This will help you. और ये बर्डिंग ईस्ट में डिफरेंट केस है. CDK is common in budding yeast. CDK1. Cyclines are different. This also is easy to remember. How? 3, 2, 1 and then 5, 6, 4, 3, 2, 1. Okay, ये numbers remember. And this is first of all CLN, right? CLN, and this is about CLB. First 3, then 2, 1. Okay. D का counterpart है, and this is of E. This is A, and this is B. Counterpart है in budding yeast. You can see here the written जो cyclin D है इसमें 3 cyclins है, D1, D2, D3. And original name of CDK1 was CDC2 in vertebrates and fission yeast, and CDC28 in the budding yeast. Okay, small information from this single picture is very important actually, right? So questions बनते हैं यहाँ से. The single picture chart can answer question. Okay, about phases, cycling, CDKs, counterparts in yeast. तो इसका आंसर क्या होगा? CDK2 is basically dissociation of inhibitory subunit, no. Association with cyclin A, yes. Right now, association with cyclin A. Inactivation of CAK, this is cyclin activating kinase. Activity of B1 kinase, no. The direct question from that table you can answer. Next question, which one of the following options best represents cholesterol distribution as a percentage of total membrane lipid in typical animal cell? Okay, cholesterol distribution का percentage. Now, if you don't know this, I will answer straight away and remember this. Okay, cholesterol का high percentage is found in plasma membrane. Okay, then lysosomal membrane. Okay, lysosome we know वहाँ पे degradation होता है, right? Then smooth ER. Okay, smooth ER which is responsible for lipid synthesis, right? So cholesterol इसमें होगा. And then last, inner mitochondrial membrane. Okay, so option 3 इसका आंसर है. Got it? Just remember this. This is a direct question. Which one of the following features differentiates bacteria from archaea? And this is also very repeating question. Okay, bacteria versus archaea बहुत बार पूछा है, तो इसका भी टेबल है. I will show you these tables. Okay, very important. Make a screenshot, right? Differences are asked. Okay, so bacteria में peptidoglycan is there, which is not there in archaea and eukarya. Okay, and ये जो next point है ना, membrane lipids का, this is also very important इसमें, right? यहाँ पे ether linkage है in archaea, in bacteria it is ester linkage, as well as in eukarya ester linkage. Got it? Then here it is methionine in archaea and eukarya, bacteria में formyl methionine. हमने देखा है molecular biology में. Antibiotic sensitivity in the case of bacteria, not found in the archaea and eukarya. Okay, what else? See the things, right? I will not extend. One more table. See, this is more detailed. Okay, so questions from these tables are asked basically many times, right? And this is very helpful. So now इसका answer, presence of a cell wall made up of peptidoglycan. Yes, first option ही answer है. Right. Absence of nuclear membrane, दोनों में है. Nuclear membrane एब्सेंट इन बोथ बैक्टीरिया एज वेल एज आर्किया, तो ये नहीं होगा. प्रेजेंस ऑफ सिंगल सर्कुलर क्रोमोसोम, दिस इज ऑल्सो फाउंड इन बोथ, तो ये भी नो आंसर. दिस इज कॉमन. प्रेजेंस ऑफ मल्टी सिस्टोनिक ओपेरॉन, कॉमन इन बोथ. पॉलीसिस्टोनिक, राइट, single promoter many genes, so these are common. Only the first is peptidoglycan presence seen only in bacteria and not in archaea. Next question. All of the following are covered by lipid monolayers except matlab we have to identify that option that is having double membrane. Okay, the three options are having monolayer. So lipid droplet monolayer. Okay, monolayer is single layer, right? Single layer means what? ऐसा. And double membrane means we know like this. So double membrane. तो हमने प्लाज्मा मेम्ब्रेन देखा है. And then that plasma membrane का double coiling like this. Okay, this is double layer. तो लिपिड मोनोलेयर लिपिड ड्रॉपलेट में है VLDL, very low density lipoprotein, monolayer. Endocytic vesicle, double membrane. Okay, this is a double layer. Three option है, right? Chylomicron, again monolayer. So endocytic vesicles ये प्लाज्मा मेम्ब्रेन से डिराइव्ड है ना. We know these are endosome, basically endocytosis जो होगा, like this is a membrane and इसका ऐसा होगा and then it will get separate like this. So this is a vesicle. Okay, so it is having a double layer. Next question. Which one of the following enzymes can unwind short stretches of DNA helix immediately ahead of a replication fork? Okay, see this is a very tricky question. अभी यहाँ पे क्या लिखा है? Unwind, right? Unwind. So you will think helicase will be the answer, right? Helicase answer होगा because helicase does the unwinding. Okay, we know this. Unwinding में helicase का role है. But here the important term is immediately after a replication fork. Now see, this is a DNA, this is a replication fork, right? यहाँ पे helicase ने unwinding किया है, but यहाँ पे अभी नहीं किया है. So here after the replication fork, just ahead, जो एंजाइम काम करता है for unwinding, first that is DNA gyrase. Okay, DNA gyrase answer है. Now DNA gyrase and topoisomerase are also same. We know that DNA polymerase तो unwinding में नहीं होगा. Polymerase का काम polymerization है. But why not topoisomerase then? And why DNA gyrase only? So basically, what is the role of topoisomerase? Topoisomerase का role क्या है? Prevent supercoiling, right? So when the helicase acts, first helicase acts to separate the strand, which causes supercoiling. And वो supercoiling को कम करने के लिए DNA gyrase यूज होता है. So DNA gyrase is basically topoisomerase 2, right? Topoisomerase 2 होता है, right? And this is why the topoisomerase is not the answer, because if we say topoisomerase, then topoisomerase 1 भी आएगा and 2 भी आएगा, but we want only 2 as the answer. So DNA gyrase is basically topoisomerase 2. Okay, topoisomerase 1 cuts single strand, but topoisomerase 2 cuts double strand, right? So ये आंसर हमें चाहिए. So DNA carriage is basically topoisomerase 2, and thus that is the answer. Okay, so here the term that is important is unwind short stretches of DNA helix immediately ahead, मतलब replication fork के तुरंत बाद which enzyme unwinding करेंगे. So that answer is DNA gyrase. Now next, which one of the following statements depict the correct function of TF2B? Now this is transcription factor 2B. Okay, transcription process हमने देखा है, you will find the playlist of molecular biology, proper detailing में देखा है. So the order of basically factor binding is DABFEH, right? अभी इसका रोल सेपरेट है, तो D जो है that binds टाटा बॉक्स. ओके, टाटा बॉक्स को बाइंड करने वाला D है. तो the first option, it binds to the Tata box and helps in the proper positioning of RNA pol 2. नहीं होगा आंसर. Why? Because B का रोल ये नहीं है टाटा बॉक्स को बाइंड करना. So this will not be the answer. It plays an important role in elongation phase of transcription. Elongation phase में इसका रोल है. No, इनिशिएशन में रोल है इसका. हमने देखा इनिशिएशन में ट्रांसक्रिप्शन फैक्टर्स बाइंड होते हैं, तो इलोंगेशन में नहीं है रोल. इट फॉर्म्स द ट्रांसक्रिप्शन इनिशिएशन कॉम्प्लेक्स बाय इंटरैक्टिंग विथ आरएनए पॉल 2 एंड टीबीपी. यस, ये हो सकता है, राइट? ये हो सकता है, दिस इज करेक्ट. तो ट्रांसक्रिप्शन इनिशिएशन कॉम्प्लेक्स का पार्ट है, राइट? इंटरैक्ट्स विथ आरएनए पॉलीमरेज 2 एंड टीबीपी, राइट? एंड टीबीपी इज व्हाट? टीबीपी हेल्प्स, जो डी है फर्स्ट ट्रांसक्रिप्शन फैक्टर, इसके साथ एसोसिएटेड है टीबीपी एंड टीएएफ, राइट ना? एंड बी कैन हेल्प इन द प्रॉपर फॉर्मेशन ऑफ इनिशिएशन कॉम्प्लेक्स. तो यस, ऑप्शन 3 करेक्ट हो. नेक्स्ट इज इट एक्ट्स एज अ co-activator that facilitates the binding of enhancers to the promoter. No, ये भी नहीं है आंसर. co-activator नहीं है, activator ही है. Transcription factor, it is important for initiation, right? Exact role इसका है BRE element binding. BRE is a promoter sequence. BRE element binding, BRE को बाइंड करता है for efficient transcription initiation. तो जो फोर्थ स्टेटमेंट में दिया है बाइंडिंग ऑफ इनहैंसर्स, ये रॉन्ग है. नेक्स्ट क्वेश्चन, राइबोसोम्स अटैच टू द एंडोप्लाज्मिक रेटिकुलम वाया. ओके, एंडोप्लाज्मिक रेटिकुलम अभी हमारा चल रहा है इन द सेल बायोलॉजी. तो इसका आंसर क्या होगा? राइबोसोम अटैच टू ईआर, वी नो दिस इज को ट्रांसलेशनल प्रोटीन ट्रांसलोकेशन, राइट ना? मतलब ट्रांसलेशन जब चल रहा है प्रोटीन सिंथेसिस, तभी राइबोसोम बाइंड होगा ईआर से. तो हाउ इट बाइंड्स? लेट्स सी ऑप्शंस. differing surface ribosomal protein composition that allow it to interact with polar head groups on the ER lipid bilayer. No. 5' UTR of mRNA been translated that directs the localization of ribosomes to the ER? No, 5' UTR का रूल नहीं है. Next, translated nascent chain which carries a signal allowing interaction with SRP receptor on the ER. Yes, SRP receptor का binding होता है. Right. See, this is the correct answer. Okay, this is the correct one. मतलब, I will show you diagram. See, this is a mRNA. Okay, mRNA 5' and 3'. Right. अभी यहाँ पे लार्ज सबयूनिट बाइंड है, यहाँ पे स्मॉल सबयूनिट. ओके, ऐसे प्रोटीन आ रहा है. नाउ दिस प्रोटीन इज हैविंग अ सिग्नल. ओके, सिग्नल है. सिग्नल आई विल ड्रॉ इन रेड कलर. राइट. So this signal, this is basically signal, and this signal is recognized by SRP, signal recognition particle. Okay, and then this इसका receptor है on the ER, and there this signal gets bind. Okay, मतलब ये signal है, ये SRP पर, SRP receptor पर bind होता है. Okay, so the signal is recognized when it is translated from a nascent chain. Got it? So option 3 आंसर होगा. नेक्स्ट ऑप्शन थर्ड mRNA that interact with rRNA नहीं हो. Got it? Simple. Next question. This is also very easy. If 19% of nucleotides in a genome are A, what percent are CGT? Very easy. A plus T plus G plus C total 100% होता है. Right? This total is 100. Now A दिया है हमें 19%. So as we know, A binds with T, so 19 होगा, दोनों भी 19 होगा, A भी 19, T भी 19. We can get A plus T plus G plus C is equal to 100%. So A plus T 19 plus 19 है. This will be as it is. 19 plus 19 is 38. 100 minus 38 is equal to G plus C. Right? 100 minus 38 is 62. Right? 62 is G plus C. So इसका individual will be half. Right? So 2 3s are 6 and 2 1s are 31. G will be 31, C will be 31. So basically I showed you the steps to calculate, but इसमें एक ही ऑप्शन से हमारा आंसर हो जाता है. हाउ? सी T19 एक ही ऑप्शन में है, ऑप्शन नंबर 1. ओके, T is equal to 19 present only in option 1. तो ऑटोमेटिकली बाकी सब इनकरेक्ट. राइट? यानी 31 हमने सॉल्व किया. Like this quickly you can solve the questions. Okay, now the next one is about G protein. Okay, the heterotrimeric G protein signaling component that is uniquely present in the plant lineage is. Okay, now G protein is also very important concept. Basically ये cell signaling में आता है. Okay, cell signaling अभी हमने start नहीं किया है, not discussed, but this concept is very important. Now ये जो GT phase acerating protein है present in animal as well as plant, तो ये आंसर नहीं होगा. GPCR G protein coupled receptor ये भी दोनों में प्रेजेंट है. राइट? रेगुलेटर ऑफ G protein signaling RGS ये भी दोनों में प्रेजेंट है इन मोस्ट ऑफ द केसेस. राइट? RGS, बट जो एक्स्ट्रा लार्ज G अल्फा सब यूनिट ये सिर्फ प्लांट लीनिएज में प्रेजेंट है. ओके, so this is a direct question basically, and ये important है, मतलब this you have to remember. right? So extra large G alpha subunit. G alpha subunit is basically important for G protein signaling. If I give you a rough idea, G protein is basically which binds with GTP. Okay, अभी ये प्रोटीन है GTP से बाइंड होगी, right? And this is heterotrimeric. Heterotrimeric means what? Three different subunits: alpha, beta, and gamma. So this alpha one binds with GTP, and beta gamma is separate. Okay, and this alpha and gamma ones are bind with membrane, right? Membrane से inserted है. Okay, remember these things. Basic I will be telling you here. Alpha one is membrane attached, gamma also attached to membrane. And ये alpha, when this alpha gets bind to GTP, beta gamma separates, right? Separate हो जाता है. And then this will run a cycle, okay, a signaling process. Got it? So when this is bound to GTP, it is said to be active, right? Active है. And when this is bind to GDP, this is inactive. Inactive. Got it? मतलब GTP से जब bind है तब active है, and जब GDP से bind है तो inactive. Okay, so that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Like this video, share this video with your friends, subscribe to our channel, and stay tuned for the upcoming videos. Thank you.